In today's lecture, we will explore how cells communicate with each other, ensuring their proper functioning. We will cover various topics such as epigenetic regulation, autophagy, metabolic signaling, and more. Get ready for an exciting dive into the intricate world of cellular signaling. Epigenetic regulation modifies gene expression without changing the DNA sequence. It involves DNA methylation, histone modifications, and chromatin remodeling and affects gene accessibility. For instance, DNA methylation silences genes, while histone modifications can either activate or inhibit gene transcription. Epigenetic regulation is important in development, cellular differentiation, and disease progression. Autophagy is a crucial cellular process for cell survival and maintaining balance. It involves breaking down damaged organelles and proteins using lysosomes. This process is activated during stress or damage, forming autophagosomes that engulf cytoplasmic components. These autophagosomes then fuse with lysosomes, leading to degradation. The resulting breakdown products are recycled to support cellular functions. Dysregulated autophagy is associated with diseases such as cancer, neurodegeneration, and metabolic disorders. Non-coding RNAs, NCRNAs, are RNA molecules that don't make proteins but control gene expression. They act as molecular switches, decoys, or guides to regulate protein synthesis or chromatin organization. Examples include microRNAs, miRNAs, long non-coding RNAs, uncRNAs, and small interfering RNAs, siRNAs. Dysregulation of uncRNAs is linked to diseases. Metabolic signaling refers to the communication between cells and tissues to maintain energy balance and respond to metabolic demands. It involves the integration of various signaling pathways, including hormones, growth factors, and intracellular metabolites. These signals regulate metabolic processes, such as glucose uptake, lipid metabolism, and energy production. For instance, insulin signaling pathway ensures glucose uptake in response to high blood sugar levels, while AMP-activated protein kinase, AMPK, pathway activates energy-producing pathways during times of energy depletion. Dysregulation of metabolic signaling can lead to metabolic disorders such as obesity and diabetes. Mechanotransduction is the process by which cells convert mechanical signals into biochemical responses. It involves the detection of mechanical forces, such as tension, compression, or shear stress, by cells and the subsequent activation of specific signaling pathways. Mechanotransduction plays a crucial role in various cellular processes, including cell migration, tissue development, and response to mechanical stimuli. For example, integrins, transmembrane proteins that mediate cell adhesion to the extracellular matrix, can activate intracellular signaling pathways in response to mechanical force, leading to changes in cell behavior and gene expression. The temporal control of signaling events is essential for precise cellular responses and coordination of complex processes. Signaling events are tightly regulated in time, with specific molecules being activated or inactivated at specific times in response to various cues. This temporal control is achieved through a precise sequence of signaling events, including ligand-receptor interactions, intracellular signaling cascades, and feedback mechanisms. For example, during embryonic development, sequential activation of signaling pathways guides cell fate determination and tissue patterning. Dysregulation of temporal control can lead to developmental defects or diseases. Membrane receptor clustering refers to the organization of cell surface receptors into clusters or microdomains, which play a crucial role in signaling specificity and efficiency. Clustering can occur through receptor-ligand interactions, receptor oligomerization, or the recruitment of scaffolding proteins. Clustering enhances receptor signaling by promoting the proximity of signaling components, enhancing signal amplification, and preventing signal crosstalk. For instance, receptor tyrosine kinases, RTKs, form clusters upon ligand binding, leading to the activation of downstream signaling pathways. Disruption of receptor clustering can impair signal transduction and contribute to disease development. Signaling in neural networks is vital for proper brain function and communication between neurons. Neurons utilize diverse signaling mechanisms to transmit electrical and chemical signals, allowing for cognitive processes, sensory perception, and motor control. One important example is synaptic signaling, which involves the release of neurotransmitters from the presynaptic neuron, their binding to receptors on the postsynaptic neuron, and the subsequent propagation of the signal. Dysregulation of neural signaling can lead to neurological disorders, such as Alzheimer's disease, Parkinson's disease, or epilepsy. Excitable cells, including neurons, muscle cells, and some endocrine cells, possess the ability to generate and propagate electrical signals. Excitable cell signaling relies on specialized structures, such as ion channels and ion pumps, to regulate the flow of ions across the cell membrane, leading to changes in membrane potential. These changes in membrane potential trigger the opening or closing of voltage-gated ion channels, allowing for the propagation of electrical signals. Excitable cell signaling is crucial for nerve impulses, muscle contraction, hormone release, and sensory perception. Signal amplification strategies are essential for ensuring efficient and robust signaling responses. Cells employ various mechanisms to amplify signals, enabling them to detect and respond to low-intensity stimuli. One prominent strategy is the use of signaling cascades, where a single ligand-receptor interaction triggers a series of downstream signaling events, resulting in the amplification of the initial signal. Another strategy involves positive feedback loops, where the activation of a signaling pathway enhances its own activation, creating a self-amplifying loop. Signal amplification is crucial for processes such as immune response, development, and cellular growth. Signaling crosstalk refers to the intricate network of interactions between different signaling pathways within cells. Crosstalk allows for communication and integration of signals from multiple pathways, thereby modulating cellular responses. However, dysregulated crosstalk can contribute to the pathogenesis of various diseases. For example, aberrant crosstalk between growth factor signaling and oncogenic pathways can promote uncontrolled cell proliferation in cancer. Understanding the complex signaling crosstalk in disease is crucial for the development of targeted therapies and interventions. Organelle communication and signaling are crucial for coordinating cellular processes in eukaryotic cells. Organelles like the endoplasmic reticulum, mitochondria, and Golgi apparatus communicate through physical contacts, transporters, and signaling pathways. This exchange of molecules maintains organelle integrity and coordinates cellular functions. Disruption of organelle communication can cause dysfunction and diseases. Cellular signaling is a complex process that regulates various physiological phenomena. Our lecture covered topics such as epigenetic regulation, autophagy, non-coding RNAs, and metabolic signaling. Understanding these intricacies is important for understanding diseases and developing new therapies. We hope you now have a comprehensive understanding of cellular signaling.